My name is Jeff Hunt. I'm the Public Outreach Coordinator for the American Society for Biochemistry and Molecular Biology.
And I am Raj Mukhopadhyay, ASBMB Science Writer.
So this is a new series that we're trying called ASBMB Live, and where we are talking with experts in different fields about their scientific research and how that goes between the lab and the clinic and the policy world. And so today we have a really exciting discussion on glycosylation disorders, and we have experts from all over the place. And so I'm going to have them introduce themselves, going one by one. So we'll start with Erik.
Hi. So I'm Erik Eklund. I live in Sweden, hence the dark outside here. It's in the middle of the night. And I work as a pediatric neurologist and also do some lab work on CDGs. That's pretty much it.
Great. Hud?
Well, I'm Hud Freeze, and I'm at the Sanford Burnham Medical Research Institute in La Jolla, and that's why it looks light back here, because it's the middle of the day. It's not quite 70 degrees, but it's warm enough. And I've worked on glycosylation disorders for about the last 16, 17 years, and you're going to be hearing, I'm sure, a lot more about these things.
Great. Ron?
Hi, I'm Ronald Schnaar from Johns Hopkins. I've been working in the field of glycobiology for several decades, focusing on biomedical research and understanding the roles of glycosylation.
Cool. And Vanessa?
Hi, I'm Vanessa Ferreira, and I'm president and founder for the Portuguese Association for CDG and related metabolic diseases, and I'm a sibling of a CDG patient.
So as we start talking about CDGs, please feel free, those of you who are in the audience, to jump in with questions. You can either use the Q&A widget that's on this Hangout, or you can just write your questions on the event page, and Jeff and I will make sure that the questions get to our panelists.
Cool. Everybody ready? Everybody ready to get started?
Right.
Yeah.
All right. So this first one is to the field. So can we just start off by defining what CDGs are, and how did each of you sort of get introduced to this disorder?
It's an interesting story. I was a medical student and a PhD student in Sweden, and I went to a seminar, and this crazy professor from the US is standing there, and he's talking about how he's treating kids with a simple sugar, and I thought that was really cool. I'd been in glycobiology for several years, and it was never any talk about patients, and all of a sudden this guy comes out from the US and says, I can treat patients with problems with glycosylation. And that's the first time I heard about it, and that was obviously Hud coming to Sweden and teaching us about these disorders. So, but a little thing on the disease, I think you should actually give the introduction, Hud, on what CDG is.
I got started in CDG because I worked on dictyostelium on slime molds, and we were looking at mutants in glycosylation, and many of the techniques that we used were the kind of basic science things, radioactive labeling and things like that. And then I heard about that there was these patients that had this disease, had something to do with glycosylation, and we managed to get cells from a few of the patients from a colleague here in town, and found out that some of the characteristics of those cells from patients with an unknown glycosylation abnormality looked very much like our slime molds. So basic science now all of a sudden making this transition into something that had to do with patients. And that was especially close and dear to my heart, because these patients turned out to have mental disabilities and seizures and things like this. And I have a sister who is doesn't have a glycosylation disorder, but nevertheless has these kinds of problems, so it was an immediate attraction for me. And then we began to do more work looking at how we could improve the faulty glycosylation in these cells. So that was in, I think about 1994, 95, something like that.
Cool.
Vanessa, would you like to tell us how you got introduced to CDGs?
Yes, so actually in our case we lived with the wrong diagnosis for 16 years, And at the age of 16, my sister went to an emergency in a hospital, and we got a medical doctor that recently had came from a conference in which he attended to an oral presentation from Professor Yaken. And he did the follow-up of my sister during several days, and he started to see some typical signals for CDG, and he immediately suspected that it was probably a CDG. He sent the samples to Belgium, and we got our diagnosis at the age of 16 years old.
Historically, what has been the diagnosis of CDGs? When was the first link between what we call a CDG now and glycosylation? How did that link come about?
Professor Yaken, he's the granddaddy and originator of this field. And he had two patients. He's in Belgium. Brilliant pediatrician. Diagnosed a lot of metabolic disorders and discovered them. So he had these two patients, twins, that had the same set of clinical abnormalities. And what he noticed is that as you do various kinds of clinical tests, many of these tests involve glycoproteins. And he noticed that a characteristic was that abnormalities in these proteins that would seem to have no obvious connection other than some common feature, and that common feature turned out to be glycosylation. And so he thought quite brilliantly that maybe it was something to do with abnormal glycosylation. And then rather soon after that, he was able to show that these two patients, as well as others that he began to discover, had an abnormal isoelectric focusing pattern of a common indicator protein called transferrin. It's a protein that carries iron around in the blood, and they contain negatively charged sialic acids in their sugar chains, and if you don't have enough sialic acid, you get a different isoelectric point. Basic science again. And that came to be the defining characteristic of many, many of these disorders.
So how do you define a CDG? I mean, so in the sense of, is it always something that's, is it an inheritable disorder, or, are there different classes of glycosylation disorders?
It's always inheritable, and it's almost always recessive, which means that you need at least one bad gene from mom and one bad gene for dad. There are some instances where you can have a mutation occur, actually in somatic cells, haven't they showed? So there are other examples, but mostly it's a recessive disorder. And it always affects N-glycosylation, the way we define it today. It can affect other types of glycosylation as well, but the way CDG is defined right now, I would say it must at least have a deficient N-glycosylation. But there is still debate on how to define that. Well, what do you other guys say?
I wouldn't particularly agree with that point of view. I think that congenital disorders of glycosylation capture all kinds of glycosylation, and in HUD's continuing collection of year-by-year number of CDGs that have been identified, he does break those down into N-linked and precursor and glycolipids. And so I think it's broadened now. For those who might not be as familiar, we think of three basic kinds of glycosylation: protein glycosylation, which can be N or O, proteoglycans, and glycolipids. Some of the disorders that we're talking about are actually can be in metabolism or processing of precursors to all of those. So it may be affecting all of those. Others are very specific for one or the other.
Well, I'm not sure that the alpha dystroglycanopathy people would agree that they have, not all of them agree that they deal with CDGs, This is still, there is a fight. I mean, the original definition was at least N-linked is down. And now we know that many other pathways can be affected, and we call them human glycosylation disorders, which is totally fine. But I'm not sure everyone would dare to agree that it's a CDG.
Yeah, we have this discussion very often because you can imagine that anything that affects glycosylation in any pathway could be considered a congenital disorder of glycosylation. It's not outrageous. And yet we already know that it's called galactosemia or fructosemia or Walker-Warburg syndrome. So for us as practitioners, you might say, and biased glycobiologists, to say, okay, all you sit down, we will now call all of your diseases that you've worked with over the decades, we're going to call them CDGs. You know, it's a little bit sensitive, so we try not to dwell too much, you know, on nomenclature, but certainly we kind of embrace all of these. And if you would, Jeff, I don't know if you can put slide number 3 on just for a second, just to give people a sense of what we have in terms of the number of kinds of different disorders and the way those have grown over the years.
Raj, can you pull that up? I'm not having trouble sharing.
Yeah, it's not cooperating.
Oh.
That's
You know what I can do? While you keep talking, Hud, I can post the slide to our event page, so you keep
Okay,
talking.
all right. Well, the bottom line is that due to, I think, a greater and increasing awareness of glycosylation disorders, that there have been more physicians and more scientists who have gradually become involved. And, you know, about, what, 15, 18 years ago, there were just a couple disorders known. And now, as an aggregate, there's 103. And we've got several more that will be coming out in the coming months. So this is the most rapidly expanding area of identification of new kinds of disorders that fall into a category that has been maybe not well studied by traditional biochemists and certainly not a lot of physicians.
I think it's important to point out that the transferrin test will not catch all of these. It will only catch the ones that have an infection in end-linked. And many physicians think of CDGs as something you can test with a transferrin test. So we will have to do a lot now to increase of the diagnostic number of tests to do, like GPI anchors and you gag disorders and so on.
Can you explain what the transferrin test is?
That's what Hud told about the first patients that Jaken had. He found out that transferrin, which is a protein in serum that binds to iron and carries iron, carries end-linked chains, so carries one type of glycosylation. And when you have a CDG, it does not glycosylate properly. But that is only for end-linked glycosylation. And many doctors know that this is the test for CDG, so that's why it also gets troublesome for them to comprehend that everything now could be referred to as a CDG, I think.
Well, so I guess one question that I would have is, given the prevalence of glycosylation in the genome, how many proteins are associated with it, does the number of identified CDGs underrepresent what's possibly out there? Is it? I mean, are you surprised that there aren't more of these?
We think there are many more, and we just don't find them because we don't look for them. Problem is that when CDG came around, almost every patient was a type 1A or a PMM2 CDG, and they often have a classical phenotype that looks the same, and they have inverted nipples, and they have funny fat pads, and if you look like that, clinicians will find you. But if you don't look like that, people don't look for you. Doctors don't look for it. And that's something we've been advocating for a long time now, that they should start looking in other groups of people.
And I think, Ron, go ahead, please.
Yeah, so what I was going to say is that genome-wide linkage analysis with new generation of fast sequencing is identifying genes in these pathways as the basis of diseases that nobody really knew were related to glycosylation. And among those, and they may not be formally CDGs and N-glycosylation, are diseases, for instance, of seizure disorders that were known to be familial, intellectual disability known to be familial, and when the gene linkages come up with silyl transferases, for example, the geneticists then seek out glycobiologists and say, what's this about? And this is an opportunity for us to begin to probe the functionality of these genes, and yes, I think that there's going to be a lot more.
Yeah, I completely agree. You know, we estimate that about 2% of the genome is devoted to glycosylation. But in addition, if you consider that glycosylation, and we're talking about things that occur in the ER and Golgi, you realize that you have to have an organization within the cell that efficiently traffics the glycosyl transferases, collects them together in the right area, keeps them together, and all of the recycling that goes on, that there are possibilities for abnormalities in those trafficking pathways that can cause abnormal glycosylation. And in fact, there's a large number of those. And going forward, I think you can consider this year's Nobel Prize, Randy Sheckman and Jim Rothman, and the kinds of things they were looking at in terms of trafficking. It is not inconceivable that you're going to find that many of these proteins that are involved in trafficking will later turn up to be affecting glycosylation, because after all, they keep the ER and Golgi homeostasis balanced. And so with this, there are going to be a number of disorders coming along that will show abnormalities in glycosylation. Now, the thing we don't know is that particular glycosylation abnormality causal for the symptoms? Well, we don't know that. But what Ron said in terms of discovering with whole exome and whole genome sequencing, and we've done many of these, where you discover new disorders. And in some cases, we knew about abnormal transferrin, and that's why we got them. But now, in other cases that we're running into, we're finding that people who have done sometimes clinical whole exome sequencing, and there's a fee for service, are coming up with glycosylation abnormalities or mutations in glycosylation genes, and those abnormalities can be measured in some marker proteins occasionally. So I think there's a big explosion yet to come, and physicians are gradually, I think, becoming more aware of.
So I'm going to take the conversation back to how Erik started this whole conversation. He said he went to a talk of yours, Hud, and heard you say some crazy things about treating a kid with sugar. What CDG are we talking about there in that story, and how does that story with, I think it's called CDG1B, can it be an example or an anomaly in how CDGs are handled, and both clinically as well as in terms of research?
Well, I think that's a really good question. So you're right that this is CDG1B, and it was the second real disorder that was discovered. And it's a deficiency, and I don't know if you can get to the slides or not, but
we're trying.
Okay, but the main thing is we ran into this because, again, we had some cells in the lab, and we found that those cells, in this case, had an unknown defect, and it turned out it was not CDG1B. But in this case, there was a deficiency in glycosylation, so we took a simple-minded approach of saying, well, since mannose is a major component of these glycoproteins, what if you just added mannose to the culture medium? You know, really simple-minded. Well, it turned out that it improved the glycosylation. And then we went on to say, well, what if you would take that mannose and give it to patients? I mean, it's a natural sugar. It shouldn't do any harm. What if we did that? Well, so we took people out of the lab, went down to our conference room, and drank mannose for 2 weekends. And, you know, we had permission to do this. It was all FDA approved. And so all of us were drinking mannose, tapping our blood and looking at the absorption clearance rates. And literally 2 days after that study was completed, we got a call from a doc in Germany, Thorsten Marquardt. Thorsten had a patient, and he said this patient has some glycosylation disorder. We don't know what it is. But we know that you had talked about giving mannose. Do you have any idea how much mannose to give? And I said, we just finished the studies 2 days ago. Give them this much mannose, this often. Here will be the kinetics of absorption. And, let us know what happens. Well, it turned out, and I couldn't believe this, that the kid got better. And he had protein-losing enteropathy. He had a coagulopathy, so bad coagulation. He was losing proteins through his intestine. He had hypoglycemia. So all of these things went away. And of course, I thought, this is coincidence. This can't be real. In fact, we visited the little boy in Germany, and with the idea that take him off mannose. If it's really mannose, he should get. And you can imagine what the doctor said to me. You go play your lab games, son. We're keeping this kid on mannose because this is as good as he's ever been. So the bottom line is we figured out, after going down to the beer garden and thinking about it for a while and seeing some results we had on his cells, that this patient was deficient in phosphomannose isomerase. And that interconverts fructose 6-phosphate and mannose 6-phosphate. And what happened was that there is an alternate pathway to making mannose 6-phosphate directly from mannose. So he was basically taking mannose from the bloodstream, incorporating it into the cells, making glycoproteins and correcting the defect. Now, is that something that we can know is going to work for lots of patients? No, but we did get some sort of a fundamental science perspective out of it. And what we realized is that if you even move a metabolic flux, and that's what we're talking about, the sugar going through the pathway, even if you move this a little bit, because we only increase the plasma level of mannose by 3 or 4-fold, even if you move that a little bit, that's enough to make a difference, literally, of life and death. And so what came out of it was something that goes from basic science to the clinic, and then from the clinic back to basic science, and now gets us thinking about metabolic flux. So any reaction in glycoprotein synthesis that involves mannose, at least a subset of those mutations in different enzymes might be susceptible to shoving a little more mannose through. So that's why that was really important. Again, back and forth between the clinic and the basic science.
So I think this next question, based on what you said, Hud, is actually, I think Vanessa can answer this one. Hud talks about the connection between the lab and the clinic. What is your group, how does that fit into that relationship between the lab and the clinic, and what happens with patients?
Well, actually, we are lucky in the sense that we have a very committed community of scientists and medical doctors, and together we are boosting awareness for CDG, which was already mentioned by Hudson, Fries, and Erik, that is a need to do awareness. And our activities, they are basically focused on awareness activities, information, education, and empowerment. Empowerment not only for families, but as well for professionals, because in the professional environment, the medical doctors, for example, it is very important to promote events like conferences that we organized last September, and this conference, it was the first world conference on CDG. We had 72 professionals worldwide coming to Barcelona to join almost 50 families. So this is the proof on how we can work together and boosting awareness and visibility and recognition for congenital disorders of glycosylation. Unfortunately, at this point, we did not manage yet to fund research, but it is our goal, because we know that if we want to accelerate translational research, a way is giving funds and being a partner in the projects from our researchers.
Cool. So
I hope the others agree with me that we are in the good direction of boosting awareness. Maybe Hudson, you can tell us a bit about your feelings in Barcelona.
Oh, it was terrific. It was so enjoyable to see Vanessa take the reins and drive this meeting forward. She was absolutely terrific. She did a marvelous job of organizing this, of bringing the families together from Europe, from the U.S., some from Australia, and having a mix of being able to have physicians talk to scientists, talk to families. And I think Vanessa probably agrees with this, that this can be the future of how we deal with rare diseases. You know, we do this for glycosylation, but of a paradigm that we can use as biochemists. As new diseases are discovered, and they will be because of whole exome sequencing, we can use this kind of a platform of interaction between families and scientists and physicians. I mean, our labs are just a click away, right? I mean, all you got to do is, my kid has this disease, look for us, there we are. So that's going to be more and more frequent, and I think the glycosylation disorders, and especially the way Vanessa has taken this on, really gives us a thrust forward. So I think that this can model a lot of diseases that will be discovered in the next few years.
And if I can add something, for example, we are now writing projects, scientific projects, for example, with Paula Videira, a professor in the Faculty of Medicine in Lisbon, that was introduced by Professor Fries, and we did an agenda focus on research for congenital disorders of glycosylation based on families' needs. And one domain is glycoimmunology, and our project that we aim at getting funds is focused on this area. So it is interesting to see how we can boost research and collaborate as partners, as writing grants, etc., with our community of scientists.
Cool. So speaking of some of those patients, we actually have a bunch of questions that have come in from some of the families. And so I'll start with the one that came in first, and then we can just sort of roll through those. So the question points out that there was an article about a sibling pair of adults that had PMM2 CDG but didn't show any symptoms, and was wondering if there was anything further that had been found out about these individuals or about this disorder.
There's a spectrum, and Erik can speak to this well as a physician. You can have the same mutation even sometimes in sets of twins and have rather different clinical presentations. And in this case, those are probably the mildest patients that have been seen. And even though they do have mutations, and mutations that are damaging and that cause a lowering of the activity of this enzyme, which is a key enzyme in mobilizing mannose for glycosylation, there's a very, very broad spectrum. And one of the patients that we worked with and have known for a very long time just sent us her PowerPoint presentation from her high school biology class, where she defines different things that one would expect somebody in a high school biology class to know. And she even points out that she has CDG and why she has it and her brother doesn't. So, this is certainly toward the mildest end of the spectrum, and you will see this. And we wish we could get all patients to that.
There is another paper coming out on a Swedish woman with PMI deficiency who has had no symptoms whatsoever. She was caught when she was 45 in an alcohol screen at work. So they screened CDT, and she was really positive in CDT. She has had no symptoms from the intestine, no symptoms from her coagulation, nothing over her whole life. And then she has an activity that is down to maybe 20–25%. which in some patients definitely cause a disorder. So yeah, it's fascinating. It might be that if we go out and just sequence people, we would find mutations that should be damaging, but they still don't have any symptoms.
The inherent question is are those people doing something different?
Yes.
that is keeping them from having symptoms, and are there any concepts that can arise from that?
It's a very good question. I mean, there are many metabolic disorders where the patients find their treatments themselves because they avoid food and they avoid stuff because they can feel that I don't feel good when I eat this. And maybe there is something like this in glycosylation disorders as well. But I think they must have very mild mutations. That's the first thing. If you have a really bad, let's say PMM2 mutation, you will be sick, whatever you do.
You know, and one of the other things I think that's important to point out is we look at these mutations and we know that in some cases there's 20-25% residual activity. And parents are 50% activity and they're normal. So where in that spectrum from 25% and you're a patient to 50% and you're not a patient, how much do you have to boost that to get into something that will be corrective? And there are companies that are working on this. A company I'm aware of named Agios actually presented at our meeting in Barcelona. They're interested in CDG because of the opportunities of thinking about metabolic flux and how that may influence things that go on with these patients. So I think, again, it's back and forth between the clinic and the lab.
Speaking of back and forth between the clinic and the lab, what's going on from sort of the laboratory side? We talked a little bit about what's going on with the clinic and the physician. So what's going on, in the field with people at the bench? What's the sort of the next step for those researchers?
I think at least part of it is going to be we're going to discover new disorders with exome sequencing. So, I suspect there's another 10 or 20 or 30 disorders that we're going to find in the next few years. A bigger challenge is to find out how that deficiency in glycosylation manifests in the symptoms that you see. And because glycosylation covers thousands of proteins, that's a hard one to know. One of the places where there's an advantage in this is in the dystroglycanopathies, and those are primarily defects in one glycosylation pathway that primarily affect one protein. Now there may be several, but there's one major protein. So it's been easier to look at and study that because it's a series of mutations in different enzymes in a single pathway that primarily modify a single protein. So that becomes easier, but the challenge is really to understand and now model these things in organisms. Zebrafish have come on. That's becoming very, very popular. And mice are quite useful. Huh?
And frog.
And frog, yes. And mice are still obvious models.
I can weigh in on that. So when you say where can we go next, certainly mouse genetics can phenocopy the human diseases. And once that's happened, then you can begin to look at ways to change the outcomes. Can you change the outcome in an animal model that's missing the same thing that the human model is? And we do have mice that have genes missing in glycosylation, and actually have studied them fairly extensively even before it was realized that there were clades of individuals that, or families that had congenital dysfunctions that were mirrored by these animal models that we are now trying to intervene.
I thought you said there's one more audience question.
Yeah, so there's been a couple of questions just talking about how to raise awareness and support for research and therapies into these diseases. So I guess I'll just go down the line and ask people if they're, what's sort of the top idea that they might have for generating more knowledge and support for these research and treatments for these diseases. So I'll start with you, Vanessa.
Well, my answer is definitely, first of all, it is very important to have a patient advocacy group. And in our case, because it is a rare disease, we are geographically isolated. So as many patient organizations for CDG we have working together, better. That's my first input to that question. Then it's true that leading a patient organization, because as you may suppose, it is a non-profit organization, it is very challenging because it is difficult to identify and set up priorities for all families across different countries and continents, and as well there are difficulties to have influence, because in our case for CDG, as many other rare metabolic diseases, getting into the government and decision makers, it is very difficult. And as well, it is difficult to advocate for a better medical infrastructures and for better access to services. So I think in order to overcome all these challenges which contribute to boost research, we need first of all to be all organized, and we need to have many volunteers engaged in the activities of patient organizations.
Ron, any thoughts?
So yes, I do have thoughts. I think that the way we move forward is by bringing the research more clearly into the clinical arena. And as the tools of glycobiology have improved, the analytical tools, the cellular biological tools have improved, that's happening. And that was evidenced last year with the release of the National Science Foundation's report on glycosylation and glycosciences. So I think that there is this building infrastructure, building knowledge base, and then building advocacy, especially in government agencies that can make serious contributions to further research.
I also would like to add that I also think it is extremely important to have knowledge shared collectively. So sometimes for families it's difficult, for example, to get information because it is quite dispersed or scattered. So I think if we have knowledge built collectively, like for example now in Spain there is a group from Dr. Mercedes Serrano and Belén Pérez Dueñas that are leading a project called EPASIBART and focus on CDG, which aims at from patients collect a bunch of information, clinical information and quality of life information that will be transformed in a natural history for CDG. And I know that they have already almost 60 families participating in this project. So the information that we will get from there, it will really boost visibility and awareness for CDG, and will give us the information needed to advance on therapies, because to advance on therapies we need to have a deep clinical description for these patients, and it has to be really collectively collected in order to have as many statistical data as possible.
Cool.
Hud and Erik?
Well, I agree on what's been said. Problem with collecting clinical data is that there are so few patients in each subgroup of CDG. We have 50 or so disorders, and we have a lot of PMM2 patients, rather many ALG1 patients, and ALG6 patients, and then we don't have many more. Well, maybe the PGM1s are coming, but we have 2 or 3 in the other groups, and that is hard to make any good clinical data on that. So I think it's very important that we as doctors go out and do our advocacy amongst other doctors to find these patients, because there are many out there and we need to find them in order to know better what each subtype presents as, and otherwise we can't start treating and knowing what is the natural history and what do we change when we treat, because we know that most of these disorders we won't be able to correct them. We won't be able to make them, we probably won't even be able to sort of reverse what has already happened, but we might stop a process so that sort of we freeze them at the time when we start treating. But then we have to find them, and we need to do natural history studies.
I want to follow up on something Erik said. That was one of the things at a meeting that involved some people from the FDA, and they said, look, for any drugs that you're going to develop, we need to know the natural history of what these disorders are looking like. And that's why the data collection is so important. And I think a good ray of hope here, beyond what Vanessa had just described in Europe, is that there is a segment, there are some laboratories at the NIH who are now moving forward to try to bring in CDG patients for a very, very thorough workup. So the only requirement would be that they are in some way diagnosed as CDG, perhaps just by transferrin, and that then they will have an organized and thorough clinical workup. And this would be open to patients even from other countries once they reach U.S. soil. So all the cost would be covered. So this is a great idea for us to be able to now get information. And part of the other interesting portion is that a reasonable number of patients in the undiagnosed disease program at the NIH, run by Bill Gall, has got a number of patients that have seemingly abnormal glycosylation. So these are going to help us with input from the NIH, awareness from the NIH, pharmaceutical companies being interested in trying to treat some of these patients. We've got a good start.
Don't think we have any further questions from the audience. Jeff, am I correct?
We've got a couple that are a little more specific. So I'd ask the people who have sort of specific questions to send us an email at asbmbio@gmail.com, and we will get those answered specifically for you from either the folks on the Hangout or from the folks who are doing the research or the clinical work.
And so with that, I think we'll close this conversation. Thank you so much to Vanessa, Ron, Hud, and Erik for making the time to join us on this Hangout. Thank you to the audience that joined in and sent us questions and listened in. This conversation will be archived as a YouTube video, and we will post it once the archived video is available, and you can watch it at a later date. So thank you very much, and if you want to know when the next ASBMB Live Hangout is going to happen, please check asbmb.org.